I first met Yvonne actually through a connection at Genentech, who recommended Yvonne to me as someone who is very, very passionate in this field, and would be a fantastic addition to the program. I remember my first meeting with Yvonne. I mean, it was, you will see when she speaks. I mean, she is truly passionate about what she does. She's enthusiastic, and she was immediately drawn into this cause and on board with helping, not just from a presentation perspective, but with also recommending other speakers, helping design panel discussions. So thank you, Yvonne. With regard to her scientific background, Yvonne is a board certified oncologist. She is the senior medical director of late stage development at Genentech Roche in South San Francisco, and leads a number of phase 1 through phase 3 immunotherapy studies in gynecological malignancies, including ovarian cancer and cervical cancer. She's also a practicing physician at UCSF, where she works hard to treat patients who are battling this disease. I think I will stop there just in the interest of time, so Yvonne has enough time to speak, but suffice to say, Yvonne has many, many, many accomplishments. Her bio is listed on the WOVA platform, so if you'd like to learn more about Yvonne, please visit it there. And with that, Yvonne, thank you so much for being here, and I will let you take it away.
Thank you, Jonathan. I'm just going to share my screen.
I guess I just want to say one thing. I know many of you may be logged in through the Whova platform, and whether or not you're using the Zoom-based one or the Whova platform in general, we are only monitoring the chat function in Zoom. So for all questions that come up during this session, please enter them using the chat function in Zoom, not through Whova.
Great.
Okay,
do you see the single screen?
We do. Yep.
Great. Fantastic. So good morning, everyone. Thank you so much for that kind introduction, Jonathan. You know, it's honestly my privilege to be invited to this forum and to be a speaker among all of the many experts who are lined up for today. So like Jonathan said, my name is Yvonne Lin. I'm a senior medical director at Genentech Roche. And I'm also an attending GYN oncologist at the University of California in San Francisco at the Mission Bay campus. So these are just my disclosures. I am a full-time employee at Genentech Roche. I will discuss the some products, both our own products as well as other industry partners' products. Some are FDA approved, others are not. This is purely for scientific discussion, and there's no promotional intent. These are the objectives of today's talk. And really, I think first and foremost, the number one priority I wanted to do was I actually wanted to take a moment to celebrate. And really, it's to celebrate and recognize all the hard work that each and every one of you, your family, your colleagues, your friends, and also mentors have poured into advancing the science and our knowledge about ovarian cancer. And it's really these advancements that have led to improvements in how we as GYN oncologists and scientists and people really can take care of women with ovarian cancer. This is also applies to women with all the other GYN cancers like endometrial and cervical cancer. But as much as it's a celebration, I think it's also a call to arms, as has been said earlier, because ovarian cancer is still a devastating disease. And it's certainly devastating for the women who are affected, but it's also devastating for the circles of people around her. I think for that reason, we must and we really can do better. I think one of the therapeutic successes that has been in cancer recently has really been in the field of cancer immunotherapy. And so today's talk, I'll start with a brief retrospective first of some of the important successes that we've seen with modern day ovarian cancer therapies. I'll then share with you some of the successes we've seen with cancer immunotherapy in the GYN malignancies or the gynecologic malignancies. And then I'll end with some reflections as to where we would go with ovarian cancer. And so with that, I wanted to start off by showing some of the timelines for the three primary gynecologic cancers. So ovarian cancer, endometrial cancer, and cervical cancer. And really, this is just so that we can reflect a little bit on how cancer treatment has evolved over the past several decades. I went ahead and took the liberty of marking the monoclonal antibodies pink, the small molecule targeted agents blue, and then the traditional cytotoxic chemotherapies remain in gray. Agents that have FDA approval are also written in red. And what I think, or at least what I hope you can see are a couple things.
Hey,
First,
Yvonne,
yeah.
can we pause 1 minute? There is a question in the chat that we're not able to see the screen.
Oh,
For everyone out there, if this is a common issue right now, can you put a message in the chat that you either are or not able? Okay, so a lot of people can see the screen. So and Steve, maybe try to go through the Zoom based platform instead of Hova. You should be able to. There should be a link that says join in Zoom, and if that doesn't resolve the problem, we can chat one-to-one. Okay, thanks, Yvonne. Sorry.
okay. Great. No, thanks, Jonathan. I got scared for that. I thought, I cannot IT sheet this. So I think there's a couple of things I hope you can take from this this timeline slide. And the first really is that I think the traditional cytotoxic chemotherapies really comprise the overwhelming majority of therapies in our toolbox as GYN oncologists for decades. And it wasn't really until well after 2010 that we successfully translated our knowledge about cancer biology into an actual therapeutic strategy for any of our GYN cancers. And you can see this because the first sort of pink and blue flag doesn't really consistently appear until about the 2014 timeframe. I say consistently because you can actually see way back in the early 1970s that megestrol acetate, received FDA approval for metastatic and recurrent endometrial cancer, which is logical because even back then we knew that that was a hormonally sensitive disease. But consistently, this didn't occur until well into the 2010s. And I think the second thing that I hope you can sort of see from this timeline slide is that the drugs that are based on the biology of the cancer cell, or the so-called targeted agents, if you will, really lead the way for drug development, particularly in this past decade. And so one of the earliest therapies of this type, bevacizumab, targeted angiogenesis. And you can see from these Kaplan-Meier curves that supplementing a chemotherapy regimen with an anti-angiogenic therapy led to a pretty sizable reduction in the risk of progression and recurrence, at least for ovarian cancer and cervical cancer. And then even in a couple clinical scenarios, namely platinum-sensitive recurrent ovarian cancer and cervical cancer, this addition of bevacizumab to the chemotherapy backbone went so far as to even reduce the risk of dying. Now, if you look at the years in which these studies were published, and that's it's on the left-hand side of the slide, you'll see that these publications are already about 5 to 10 years old. And so while these 30 to 50% reductions in risk for recurrence and progression were very meaningful and continue to be very meaningful and important, there's still a lot of room for improvement. And that's actually been what's happening in the past several years. A recognition of one of the key cellular and molecular vulnerabilities of certain ovarian cancers, as well as a small molecule drug that happened to also capitalize on this Achilles heel, so to speak, really transformed the landscape of ovarian cancer therapy. And of course, I'm talking about the PARP inhibitors here. So although this is the Antibody Society, I think this audience is nevertheless acquainted with the profound efficacy of PARP inhibitors. One of the early studies, Study 19, gave us some of the earliest clinical data on the efficacy of PARP inhibitors in susceptible tumors with BRCA mutations. But it's really the collective pivotal phase 3 data, which I show here, that starts with SOLO-1 on the left-hand side of the slide, followed shortly after by PAOLA-1, which is on the top row of curves, and then PRIMA, which is on the bottom row of curves. This trio of studies really transformed the treatment landscape for ovarian cancer. And I say they transform the patient experience also because you can see here that adding the PARP inhibitor to the patient's treatment regimen after they finished chemotherapy delayed ovarian cancer from recurring, particularly those patients whose tumors harbored that particular molecular vulnerability. the BRCA mutation, or a deficiency in homologous recombination. Now, although we're able to kind of unbend the survival curves a little bit with the PARP inhibitors and lift them up off the horizontal axis, you can see that patients still unfortunately recur, particularly those on the far right-hand side of the slide whose tumors do not actually harbor these mutations or these deficiencies. And so there's still a significant room for improvement. So while we in the GYN field were looking at ways to exploit this molecular vulnerability of ovarian tumors, a greater appreciation for the interplay between the immune system and cancer was developing in the greater cancer field. And this, of course, coincided with the development and the refinement of some of the contemporary cancer immunotherapy or CIT agents. Melanoma served as the vanguard malignancy for CIT. And what was notable about melanoma was that for a disease where the outcomes were uniformly recurrence and death, and unfortunately in rather short order, the contemporary immune therapies, they managed to actually utterly flatten and kind of unbend the curves as time went on. Now, I suspect that nearly everybody in the audience recognizes the gentleman in the bottom picture of the slide. And this is, of course, our 39th president of the US, Jimmy Carter, who was diagnosed with metastatic melanoma to the liver and also to the brain. He underwent multimodality treatment with surgery, radiotherapy, and the anti-PD-1 agent pembrolizumab, then later announced in 2015 at the young age of 91 years old that he was cancer-free. And it's this kind of durable response and even the elimination of cancer, particularly melanoma, that has already metastasized to the brain, that is really considered a clinical game changer in the field of cancer therapies. And it justifies its earlier award designation as the breakthrough of the year. Now, while melanoma served as one of the earliest testing grounds for cancer immunotherapy, virtually every other human cancer rapidly followed. Everybody was eagerly hoping to replicate the successes seen in melanoma. The hypothesis that activating or reinvigorating the immune system could lead to enhanced tumor cell kill was successfully tested in multiple clinical trials across multiple tumor types over the past several years. And this has resulted in a now sizable body of evidence establishing PD-1 and PD-L1 therapeutic blockade as a valid and efficacious therapeutic strategy for many tumors. This also includes tumors from the GYN track that are outlined here in blue. So let's take a deeper dive into what we've seen with immunotherapy and some of the gynecologic malignancies specifically. The genomic interrogation of endometrial tumors, fueled in large part by the ongoing TCGA or the Cancer Genome Atlas project, revealed that a fraction or about 30% of endometrial tumors carried a deficiency in DNA mismatch repair. So this highly conserved biological pathway is important in maintaining genomic stability. And therefore, defects in this pathway increase the spontaneous mutation rate for those cells. And that's reflected in the graph in the upper left corner. Here, the red box highlights the uterine cancer cases, and you can see that there's a select group of tumors at the very top of that red box with the speckles that have an extraordinarily high number of somatic mutations. This increase in the mutational burden was hypothesized to be associated with increased immunogenicity and therefore further hypothesized to be more sensitive to the immunomodulatory approaches offered by cancer immunotherapy treatment. Well, this hypothesis panned out in systematic clinical trials. So here in the middle panel and also on the right panel, you can see the waterfall plots on the top and the swim lane plots on the bottom, in which the anti-PD-L1 molecule avelumab and the anti-PD-1 molecule dostarlimab were tested in endometrial tumors that harbored this particular molecular vulnerability. And what it showed was that tumors that have this defect showed not only amazing shrinkage in the tumor size when exposed to, or in other words, treated by these specific CITs, but in addition to that shrinkage, the duration of tumor control was also much longer than what we would have expected with just standard of care alone. Now, what about other GYN cancers? Well, some cervical cancers also appeared to have the same kind of sensitivity to this kind of immunomodulatory approach to treatment. What's different with cervical cancer compared to endometrial cancer, though, is obviously the underlying biology of these two tumors. Cervical cancer, as most of us know, is largely, if not uniformly, virally driven. HPV, or the human papillomavirus, is seen in nearly all cases of cervical cancer, with chronic HPV infection actually associated with increased expression of PD-L1. And you can see in the left-hand panel, and as well as in the middle panel, treatment with the anti-PD-1 agent, be it pembrolizumab or balstilimab, both led to shrinkage of tumor size. And many of these responses also lasted a significant amount of time, longer than what we would have expected based on historical evidence and the historical experience. So, in fact, this immunomodulatory approach was then tested in a direct head-to-head comparison against standard of care chemotherapy in this randomized phase 3 clinical trial. And in this trial, the Empower Cervix 1 study, cemiplimab showed remarkably better tumor control and ultimately a survival benefit over typical standard cytotoxic chemotherapy agents. What was also shown was that this approach targeting PD-1 might actually be more potent for those patients whose tumors overexpressed PD-L1, and that's shown in the lower right panel. But now, even though this data that I'm showing here is brand new from data that was released and announced this year, it's clear that there's still significant room for improvement for what these women with these particular GYN cancers. And so how would we go about doing that? Well, to understand how we can improve upon the anti-PD-1, PD-L1 agent's efficacy, I think it's worth anchoring back to this cancer immunity cycle, which I think many of you are well familiar with. Now, it's been almost a decade since this paradigm was put out there, but this paradigm really forms kind of a framework for understanding how a patient's immune system responds to cancer. So this would encompass steps from the very, very beginning with tumor-associated antigen recognition by the immune system, processing and presentation, all the way to the moving clockwise to the right-hand side of the diagram, whereby the immune system is now activated and elicits the tumor cell kill. But what's actually more important, I think, about the steps in this diagram is that it provides a framework and it highlights multiple areas for key therapeutic opportunities to combine therapeutic approaches and even agents. And you can see some of the best studied ones here with the theoretical step at which it would happen. So if you'll recall, one of the earliest modern day treatment approaches in gynecologic cancers that I had mentioned earlier was the anti-angiogenic or anti-VEGF therapeutic strategy. And there's a sizable body of translational research that shows that anti-angiogenic agents help to normalize the typically disorganized nature of tumor-associated vasculature and thus promote better T-cell infiltration into the tumors. So hypothetically, could combining these two approaches actually augment the anti-tumor activity that we already see with single-agent immunomodulatory therapy alone? And indeed, this was actually seen in endometrial cancer, where the combination of the anti-angiogenic agent lenvatinib and pembrolizumab led to notably more patients experiencing tumor shrinkage. And again, this shrinkage lasted much longer than what would have typically been expected with traditional agents. Earlier, I had shown that the immune checkpoint inhibitor pembrolizumab could do this in just that fraction of tumors who had that molecular vulnerability with the DNA mismatch repair defects, and because that predisposed those tumors to accumulate mutations. But now, combining these two approaches, the anti-angiogenic approach plus the anti-PD-1 approach, more tumors could actually become essentially susceptible to this therapeutic modality, and therefore more tumor cells could be killed. Now, this combination approach was so effective that when it was compared again in a head-to-head manner against standard of care chemotherapy, the combination of lenvatinib and pembrolizumab prevailed, which showed a significant reduction in the risk for progression or recurrence, and even a reduction in the risk of death. Again, well, what about other GYN cancers? So again, anchoring back to the cancer immunity cycle, we can see other potential combination opportunities, such as that with chemotherapies, that might further augment the anti-tumor efficacy of immune checkpoint alone. Recall that with our virally driven cervical cancers, we saw that immune checkpoint alone was able to shrink or even eliminate some tumors and even extend survival beyond what standard of care chemotherapy could offer. What if the immune checkpoint inhibitor was combined with chemotherapy? Could that whole cocktail be better than just standard of care chemotherapy alone? Well, this hypothesis was indeed tested in a direct head-to-head randomized phase 3 clinical trial, and you can see the results on the left side of the screen. That is, the combination of chemotherapy pembrolizumab was better than standard of care chemotherapy alone in reducing the chances for progression and recurrence, as well as the chances of dying. And then now also remember what we talked about with combining pembrolizumab with an anti-angiogenic agent. We saw that in endometrial cancer, and we saw superior benefit when the two were combined. Well, could we replicate that kind of efficacy in this particular cervical cancer setting? Now, this particular study did not explicitly ask and study this hypothesis, but some of their pre-planned subgroup analyses did provide early answers or early suggestive answers to that particular question. And that's shown on the right-hand side of the screen. Specifically, that in this cervical cancer study, patients who also got the anti-angiogenic agent bevacizumab in this case appeared to fare better clinically in terms of recurrence and risk of death than those who did not get bevacizumab within their cocktail. So here in these graphs, using these CIT approaches, we're starting to see that the tail ends of the survival curves are lifting up and getting farther away from that horizontal axis. But what about other strategies to further augment this immune response? Well, what about targeting the front end of the immune response? In other words, the priming or activation end, and combining that with the anti-PD-1 therapies. So that particular combination also seems promising, at least in cervical cancer. Here was the data that we saw from two separate studies. Each study simultaneously blocked CTLA-4 and PD-1. And now, while each of these data sets is smaller than the data sets from the earlier studies that I displayed earlier, we can see that there are some glimpses of some encouraging anti-tumor efficacy, particularly with the combination compared to with the anti-PD-1 agent alone. Now recall that with the anti-PD-1 agent alone, the response rate was in the 13 to 16% range. And you can see here that the combination of ipilimumab plus nivolumab shows response rates in the 25 to 30% range. This is also seen in the combination of balstilimab and zalifrelimab. Now, it's important to acknowledge that these studies are also much smaller than the ones I presented earlier, and so it's important to be optimistic, but I think we also need to be appropriately tempered with our enthusiasm until more data become available. So I've shown all this exciting new data about the immune system, therapeutically modulating it, et cetera, for GYN cancers, but I haven't mentioned ovarian cancer quite yet. And so really, is there a role for cancer immunotherapy in ovarian cancer? Like I said before, the role of the immune system in human cancers has long been recognized as being relevant. And so yes, that is the case even in ovarian cancer. You can see here on the left-hand side of the slide, this meta-analysis, which shows a pretty compelling association of the number of tumor-infiltrating lymphocytes in a tumor and likelihood of death. And basically it shows that patients whose immune systems are more engaged, so to speak, fare better than those whose immune systems are simply just not as engaged or even absent. And similarly, you can see on the right-hand side of the slide, the Cancer Genome Atlas interrogation for ovarian cancer makes a similarly compelling case for needing an immunoreactive subtype for better outcomes. So therefore, seeing that a more engaged immune system is associated with better tumor control in ovarian cancer, it would make sense, or it would be logical to hypothesize that we can therapeutically make the immune system more engaged or make it more potent, and thereby deliver an even more efficacious control of the tumor. And that hypothesis was tested with some of the early PD-1, PD-L1 single agent data showing some anti-tumor activity in ovarian cancer. Now, these response rates themselves are also quite modest, and you can see by this table. And many people also focus on the additional indicators of clinical benefits, such as the duration of response or the disease control rate. And many people anchored back to the combining those familiar nodes on the cancer immunity cycle to try to augment this single agent activity. And that really launched a number of phase 3 trials. But unlike what we saw with endometrial and cervical cancers, the combinations of chemotherapy and immune checkpoint therapy was not better than the standard of care chemotherapy and standard of care chemotherapy bevacizumab alone for ovarian cancer patients. This unfortunate surprise observation was reported out not just in a single randomized phase 3 study, but rather it was shown across 3 separate phase 3 studies, 2 in the frontline setting and 1 in the platinum-resistant recurrent ovarian cancer setting. So what's next then? I think that while that particular combination strategy didn't pan out in ovarian cancer, the underlying principles behind this cancer immunity framework still have significant merit and still point out additional therapeutic opportunities for ovarian cancer. So let's take a look at these first 2 steps. Again, the front end and the back end of the cancer immunity cycle. So Energy GY003 study tested the hypothesis as to whether or not combining therapeutic targeting of CTLA-4 and PD-1 could be more potent than an immune checkpoint alone. And as you can see from the middle column of graphs, that this was indeed the case with better tumor control with the combination of ipilimumab and nivolumab. Now, if we think about how CTLA-4 works and how PD-1 works, combining these 2 approaches makes sense. You can see that CTLA-4 targeting may actually be particularly good in a disease setting in which more cells or more immune cells need to be recruited. Or in other words, we may need to expand the clonal diversity of those immune cells so that they can attack more things. Or simply making an immunologically cold tumor hot. Now, while this data set from this particular study is quite small, I believe it does offer an early signal. And what's remarkable about the immune system is not just the multiple combinatorial possibilities that exist as outlined by the cancer immunity cycle, and also in the diagram in the upper right corner, but also there's a multitude of different targets that are offered by the different activating and inhibitory or deactivating receptors on just the T cell component of the immune response. Some of these are shown in the upper left panel. Beyond simply just the T cells, though, is actually the increasing recognition of the several other cells and components that make up the immune response and also serve as potential therapeutic targets, many of which are actively being explored, and you will certainly hear about them later today. And then finally, with the rapid advances of protein engineering, synthetic and modified antibodies can now be engineered to target multiple and increasingly specific epitopes with greater efficiency. Toxic payloads that are too toxic to deliver systematically can be conjugated to highly specific antibodies directly onto the offending cancer cells, as currently being done with the folate receptor alpha antibody drug conjugates. So in summary, I believe that in current day, immunotherapy has really emerged as a potent therapeutic strategy for major gynecologic malignancies, commonly and namely that we've seen today are endometrial and cervical cancers. However, numerous strategies exist that therapeutically modulate the immune system and further set the stage for identifying and delivering that same potent transformative therapy to women with ovarian cancer. I acknowledge that we did hit a speed bump with this first generation of cancer immunotherapy studies in ovarian cancer. But I also believe the future remains bright because we will continue to learn from these experiences. Technology will continue to advance us, and our will and our drive to succeed remains intact. We and our patients, our moms, our daughters, and our friends expect that from us. Thank you so much for attending today's symposium, and I look forward to our discussions in the network sessions. Thank you.
Thank you, Yvonne. That was a really great overview of the entire, I guess, history of treatment of gynecological cancers and ovarian cancer. That was really great. We have a question in the chat that I'll start off with because we have a little bit of time before Jose's talk. So the question from Candace is, how often are TILs in ovarian tumors not engaged? And I guess going along with that, are most ovarian tumors cold? And do you think there are ways that we can make them hot and therefore responsive to, I guess, checkpoint blockade?
So I think the first question as to whether or not do I think ovarian tumors are cold is immunologically cold. The current, I think the prevailing hypothesis of belief is that indeed they are. And that's part of the reason why, that's a big part of the reason why they haven't really, that why the first generation or the first suite of studies didn't really show that transformative benefit. The question as to what percentage of them, or whether or not we can convert a cold tumor into a hot tumor, I think that that's a great question, and I think that that is actually the focus. That's sort of the next focus of cancer immunotherapy, and sort of the next. I think the next generation of studies is really going to be how do we convert these tumors and make them hot. Jim Allison, gave a really nice summary of this at SITC last year about sort of, making his case for CTLA-4 as a mechanism for achieving that kind of a response. And, and we saw a little glimpse of that in the NRG GY003 study. So I think that, the future, I think that the definitive answer is unknown at this time, but I think that there's great optimism and curiosity to interrogate that.
Great. I guess just okay. Actually, I'll ask another question that just came in, and this actually might come up for maybe a better talk to defer to Jose. But can you speak about the role you see for engineered T cells in ovarian cancer, whether they be CAR T cells or, I guess, TILs or TCR modified T cells? Particularly in the context of the relatively low mutation rate in ovarian tumors, and why immune modulators on their own may not be working.
Yeah, I think the role of engineered T cells is fascinating because it's, and I think that there's, the challenge with ovarian cancers is that there is generally not a single defining immunogenic antigen that can be that the engineered T cell can anchor to. That said, however, there's, I think, a significant amount of work by many people in industry and academia and think tanks alike, looking to see whether or not there is a predictive algorithm, or whether or not there's some sort of learning that can some sort of machine learning or AI learning that can happen to basically identify epitopes or perhaps a suite of epitopes that might be relevant in ovarian cancers. And so I think that the current state of engineered T cells in ovarian cancer is very nascent currently, and it targets a very narrow and a very well described, very well defined patient population. But I think sort of beyond that, the next steps would be to think of creative ways to expand that kind of application in order to deliver that particular kind of cell therapy.
Yeah. I guess along the lines of that, I mean, there are a number of somatic hypermutations that occur in ovarian cancer. I mean, you touched on some of them, right? BRCA in general. Are any of these presented as neoantigens? Somewhat like the case of like KRAS, right? Mutations in KRAS can be targeted by, can be specific neoantigens for personalized T cell therapy. Does the same hold true in ovarian cancer with genetic mutations? Or do we not know yet?
Yeah, so I don't know. I'm not about that specific genetic mutation, but there are there's a number of vaccines targeting actually. So CA-125 is one, and I think later on there's I think Allison's talk is going to go over some of the antigens that have been identified as a therapeutic candidate for ovarian cancer. So maybe we can go over that during the panel discussion for that section.
Yeah, I guess I was wondering more, like if a patient has a mutation in a specific gene that's somatic and in the tumor, do we know, are any of those happen to be specifically that peptide with that mutation presented on the surface by MHC, and then therefore maybe making those patients good candidates for personalized T cell therapy, personalized adoptive T cell therapy?
Yeah.
Do we not know yet?
I don't think we know that yet.
Okay. Great. We do see a question here about C3 bispecifics in ovarian cancer, but I agree. I think we should tailor that one for Allison. She will be talking about targeting mucin with and CD3 bispecifics in her talk. And then another question from Candace just came in, and yet you addressed this a bit, but do ovarian cancers normally have high mutations?
So interesting. So they generally don't have a high tumor mutational burden. And so there's been sort of this is actually a very actively evolving sort of body of literature, and multiple groups are working on sort of collecting and presenting their data. And so at the SGO annual meeting earlier this year in February, the Imagine 050 team, Dr. Landon from UVA presented the tumor mutational burden for the ovarian tumors that were collected from the Imagine 050 study. And he actually found that the rate of the prevalence of high tumor mutational burden was actually very low, so surprisingly low. And so it kind of on one hand, it makes sense. It corresponds with sort of the response to immunotherapy that it wasn't as effective as we saw in uterine cancer, et cetera. But then on the other hand, then sort of biologically, there's some other data that sort of suggests that, maybe some of the ovarian tumors that have, defects in DNA damage repair, shouldn't those tumors intrinsically have carry a higher mutational burden? So I think what we're learning is that it's not simply just mutations, yes or no, but it's probably also the nature of those mutations that really matters in terms of immunogenicity and being able to stimulate that immune response.
Great. And I guess in the last question, I'll just throw it out there as maybe a hypothetical and speculate you can't answer, but like, I mean, what do you think? What do you think we should do? Like, what would you pick potential? Well, maybe you can't say that, but as the next best approach to trying to like push the needle on treating ovarian cancer? Clearly anti-PD-L1, CTLA-4 combinations, not super effective. Will immunotherapy ever be that effective for ovarian cancer? And if so, what do we need to modulate?
So I don't know if I agree that that CTLA-4 PD-L1 is not effective. I think that the challenge with the CTLA-4 has been that is that the adverse event profile precludes really thorough and absolute shutdown of that particular axis. And so then the question is there a way to block that particular axis? Because the purpose of that axis is to make that cold tumor hot. And so is there a way to make that cold tumor hot without that sort of global, the global effects of a hyperactivated immune system? And right now there's a lot of work with using other components of the immune system, cytokine approaches, and whether or not there are cleaner the traditional cytokines have been very global and have had very systemic effects. But is there a way to more cleanly engineer a cytokine? And I know that you are very familiar with this from your own work of synthesizing just the key parts of the cytokine in order to augment that immune response.
Yeah. So you think maybe it's not that they're not effective, it's just the tolerability is quite low. I mean, I guess we definitely know that for CTLA-4. Yeah. And so potentially engineering approaches that help us safely deliver that agent that may be active in the tumor or the metastatic or wherever, right? That's the problem with late stage ovarian cancer.
Exactly. And I also think that sort of like combining and I think that also, I'm a big believer in a lot of these machine learning algorithms, because I think that, it's very hard and time consuming, and it's the sheer number of possibilities of various conformational changes, various structures, the dynamic, even a dynamic change in conformation can elicit a completely different response, like a cis-trans kind of a situation. And so, and to have to run all of these experiments on a wet lab level is just, it's prohibitive. But I think with these machine learning approaches and AI technology, we can sort of start modeling some of these and run through some of these algorithms much faster and really be able to wet lab test the strongest, most likely candidates that have the greatest chance for success.
Yeah, that's a great segue into, hopefully the future of medicine, right? If we can actually do machine learning, artificial intelligence, and how fast can we get there? Great. Well, thank you so much, Yvonne. One other question did come in, but I think this will probably get addressed during the panel discussion. And in the interest of time, I think we will move it over to Jose. So Yvonne, thank you again for a fantastic talk.
Thank you.